Okay, good morning to everyone. The previous topic is we are discussing the what are the glands are associated inside or outside to support digestion. Now we are discussing the physiology of absorption. Physiology of absorption. Physiology of absorption. Physiology of absorption. Okay. So before we are discussing the digestion, mouth to anus, mouth to anus, mouth to anus, and also we are discussing glands. And in alimentary canal, it is having so outside glands and inside glands. Okay, mouth to anus. So what are the glands are associated outside and inside to help in the digestion, to help in the digestion. For here the physiology of absorption, physiology of absorption. For example, we take water. We will drink the water. That water will be digested? Not digested. It will be directly go through the stomach to small intestine to villi, and directly it will be absorption. Villi will be absorption. Means it is a simple form. It is a simple form. Water is a simple form, and it will be directly will be absorption. Directly will be absorption. So which material has to be absorption? Which material has to be absorption? Which material? The complex food materials. Complex food materials. Food materials. Complex food materials converted into simple absorption form. Simple absorption form. Simple absorption form. Simple absorption form. Complex food material means, so we will eat different types of food particles. Different types of food particles. That food particles are to be crushing. That food particles are to be crushing to mix with saliva to create bolus. To create bolus. To directly going to stomach. Directly going into stomach. The stomach is having churning movements. The stomach is having churning movements. The stomach is having churning movements. Churning movements. The stomach is having churning movements. The stomach is having churning movements. Means complex food materials means converted into simple absorption form. Simple absorption form. So what is that? So we will eat different types of food particles that will be crushing in our mouth, and mixed with saliva to create bolus. That bolus will be reaches esophagus to thoracic region to abdomen. It is directly goes to stomach. The stomach is having churning movement. Stomach is having churning movement, and that is called, that type of digestion is called, the first thing is, so in our mouth digestion will be started. In which type of digestion? Mechanical digestion means, so whenever we will crush the food particles to mix saliva, and small fats and small sugars and small chlorine ions will be digested, and first it is called mechanical digestion. First it is called mechanical digestion. Mechanical digestion. Mechanical digestion, after it will be go through the stomach. After it will be go through the stomach. The stomach is having so different types of gastric juices is there, and different types of enzymes is there, and it is having chemical digestion. It is having chemical digestion. Second thing is having chemical digestion. Chemical digestion. Chemical digestion. Okay, chemical digestion. Next, So we are discussing complex food materials converted into simple absorption form. So what are the complex food materials? What is that complex food materials? Complex food materials means that are carbohydrates, carbohydrates, proteins and fats. These are the complex food materials. These food materials converted into the simple absorption form. So digested by enzymes. Digested by enzymes. Digested by enzymes. This all food particles will be breakdown. It will be digested the enzymes to converted into simple absorption form. Okay. So this is first we will before we will discussing. So what are the type of digestion? This means mouth to anus. What are the associated glands will be associated to mouth to anus. So physiology of digestion will be taking the complex food materials will be converted into simple absorption form. In mouth is having mechanical digestion. Partially digested food is directly going into the stomach. The stomach is there, some churning movements is there. So the digestion will be in stomach. That is called chemical digestion. So now we will see the absorption will be started mouth to anus. The physiology of absorption. So the absorption will be start mouth to anus, where it will be how much amount of fats and proteins and carbohydrates will be absorption in mouth to anus, we will discuss clearly. Okay, this is the first thing is mouth. First thing is mouth or buccal cavity. Buccal cavity. Here the buccal cavity, the carbohydrates converted into. So who is acting here is a conversion? Tyling enzyme. It is also called amylase enzyme. Carbohydrates, another name is called starch. This is called starch. Okay, this is converted into disaccharides. Nearly 30% of starch will be converted. Disaccharides means here, what is that disaccharides? Sucrose. Disaccharides is called sucrose, maltose, galactose. Okay, in mouth or buccal cavity, the absorption, the carbohydrates will be breakdown. Nearly 30%. With the help of tyline or amylase enzyme, it will be converted into disaccharides. The disaccharide is having sucrose, lactose. And also to be releasing chlorine ions. And small fats. and small fats. Small fats are to be converted. Small fats are to be converted into fatty acids and glycerol. Fatty acids and glycerol. Fatty acids and glycerol. So, which enzyme here it is to be helping? Which enzyme? The small fats are converted into fatty acids and glycerols. Which enzyme is to be here working? Lingual lipase. Lingual lipase. Lingual lipase enzyme. Lingual lipase enzyme. Lingual lipase enzyme. Okay. So, mouth or buccal cavity, the absorption will be start mouth to anus, we will discuss. So, here the first mouth or buccal cavity we will see the carbohydrates are to be converted into nearly 30% of disaccharides. Nearly 30% of disaccharides means buccal cavity means we are already discussing. So, the mixture of saliva is releasing amylase. Amylase is called in ptyalin and also to be released as mucin and lingual lipase enzymes. Lingual lipase enzymes. Most of the time it will be most of the amount it will be released as ptyalin enzyme or amylase enzyme. Amylase enzyme going to work carbohydrates. Going to work carbohydrates. That carbohydrates are to be nearly 30% will be converted into disaccharides. Disaccharides. The disaccharides is called sucrose, maltose, lactose. Okay, and also to be releasing some chlorine ions. And the salivary gland, saliva is to be mixture of amylase is having mucin and lingual lipase. So here the small fats to be absorbed in our mouth to help lingual lipase. Lingual lipase means the small fats are to be absorption to converted into fatty acids and glycerols. Fatty acids and glycerols. Okay. Next coming to the stomach. Next coming to the stomach. In stomach is having HCl and pepsinogen and prorenin, and another thing is gastric lipase. Gastric lipase. Okay. So I will discuss clearly in stomach. The bacteria will be kill HCl. Bacteria will be HCl killed. The first action will be taken in stomach, HCl. Next, another one is pepsinogen. Pepsinogen. It is called inactive enzyme. It is to be activated to the HCl. It is called pepsin. Next, one more thing is called prorenin. It is to be activated renin. Okay, these two are inactive enzymes. Or proenzymes. Inactive enzymes or proenzymes. Okay, these are releasing for inactive stage. So whenever bind with HCl to activate these two enzymes. Okay, these two are active enzymes. These two are active enzymes. Next. So this is active enzymes. This active enzymes means renin. This active renin enzyme. This activated enzymes renin. It is going to activate. The renin going to activate. The renin going to activate means it is very important. The renin enzyme means it is very important for the children's. Very important for the children's. It is very active. It is also very active. Why? Because means the children are drinking milk. The milk is having milk casein. The milk casein is converted into calcium paracasinate. Calcium paracasinate means the milk protein is converted into curdling. The milk protein is converted into curdling. Sometimes the children are to be vomiting. Okay, so the vomiting you have to see that milk is converted into curd. The milk is converted into curd. So here the renin is very important. The renin is very important. So very important for children. Important for children. Why means the children's drinking milk. The milk is having milk casein. Milk casein. This milk casein is converted. Who is converted? Renin. The renin will be converted. The renin will be converted. Calcium paracasinate. Calcium paracasinate. It is called means milk casein means milk protein. Milk casein means milk protein with the help of calcium ions. With the help of calcium ions to converted curdling. Means curd. Converted into curdling. This is very important. Very important for children. Renin is very important. Renin is very important. So renin is important for children. So why means the milk casein is converted into calcium paracasinate. Calcium paracasinate means this milk protein is having to converted curdling with the help of calcium ions. Next. Okay, next one is called pepsin. Renin will be completed. Next we will discuss pepsin. Okay, now we will discuss pepsin. Okay, now we will discuss pepsin. What is the work of pepsin? The proteins are to be converted into proteases and peptones. Who is converted? Pepsin. Pepsin is converted. The pepsin is converted proteins to peptones and proteases. Okay, understand? Proteases and peptones. Okay, all things things are happening in the acidic medium. All things are happening in the acidic medium. Renin and pepsin. Renin and pepsin. Both are it is working in acidic medium. Both are it is working in acidic medium. Acidic medium. Okay, this is found in stomach. This is found in stomach. Now we will see the small intestine. Now we will see the small intestine. Next, small intestine. Now we will see the small intestine. Small intestine means we will see the first stomach. This is stomach. The stomach is having chyme. The stomach is having chyme. Here enter pancreatic juice and bile juice. Here enter what is this? Intestinal juice. Here enter here enter bile juice. Okay, here enter bile juice. Okay, here enter pancreatic juice. Here enter only chyme. Pancreatic juice and intestinal juice, bile juice. Okay, four are together bind the duodenum part. 4 are together in duodenum part. Finally, it will be converted into acidic to alkaline medium. Acidic to alkaline medium. It will be neutralize the food. Highly acidic medium food will be neutralized into alkaline medium. Okay, here small intestine. The first we will discuss on protein digestion. First we will start protein absorption or digestion. Protein absorption or digestion. Protein digestion. First protein digestion. Trypsinogen. First trypsinogen. Activated. Trypsinogen is activated with the help of enterokinase. Enterokinase is coming from where? Intestinal juice. Intestinal juice releases from enterokinase activated enzyme. Enzyme activator. This is enterokinase is the enzyme activator. So then activated, it is called trypsin. So by the process, okay, the trypsinogen is going to convert into trypsin. By the process is called autocatalytic process or autocatalytic mechanism. Autocatalytic mechanism. Let's start with the small intestine. The protein digestion. The first we are taking the trypsinogen. Means here intestinal so many pancreas will be releasing so many enzymes. Here it is to be activated. That enzymes will be break down proteins, that how it will be digested, okay. So trypsinogen will be converted into trypsin, by the process is called autocatalytic mechanism process, with the help of enterokinase, it is to be formed in trypsinogen to trypsin, with the help of enterokinase. It is called autocatalytic mechanism process. Once the enzyme will be activated, means with the help of enterokinase, the trypsinogen will be activated into trypsin by the process of autocatalytic mechanism. Once the enzymes will be activated, remaining all enzymes will be self-activated. Once the trypsinogen is activated, remaining all enzymes will be self-activated. Next one is chemotrypsinogen. Converted into chemotrypsin. What is chemotrypsin? Okay, these two enzymes acting proteins. These two enzymes are to be not in enzymes. It is to be converted into trypsin and chemotrypsin. It is working form. They are acting proteins. Okay. Okay, the proteins will be converted, means the proteins will be converted. Before we are discussing which enzyme, the pepsin enzyme. The pepsin enzyme will be converted, proteins are converted into peptones and proteases. Which enzyme? The pepsin enzyme. Proteins will be converted into peptones and proteases. Okay, here comes to first protein digestion we will discussion. So before the digestion will be start, means activated. Okay, the protein digestion will be started in the intestine. Okay, right. The first thing is, what is that first thing? Peptones and proteases. What is that? Proteins or peptones? Proteins, peptones and proteases. Proteins and peptones, proteins, proteases. Okay, proteases and peptones and proteins converted tripeptides and dipeptides. Tripeptides and dipeptides. Tripeptides and tripeptides and dipeptides. Are you understand? Series protease and peptones and proteins converted into tripeptides and dipeptides with the help of trypsin and chymotrypsin. With the help of trypsin and chymotrypsin with the help of trypsin and chymotrypsin. The help of trypsin and chymotrypsin. Next, and this is also proteins and proteases and peptides. They converted into tripeptides and dipeptides in the form of carboxy. And one more thing is carboxypeptidase and aminopeptidase. Okay, carboxypeptidase, three things: proteins and peptones and proteases converted. Three things are converted with the help of trypsin and chymotrypsin. The conversion of tripeptides and dipeptides with the help of carboxypeptides and aminopeptidase. Help of carboxypeptidase and aminopeptidase. Are you understand? Okay. Protein digestion. The first trypsinogen will be activated. In enterokinase. Enterokinase is coming from intestinal juice. Intestinal juice means succus centrica. It's nothing but intestinal juice. That juice will be released as enzyme activator and also releases enzymes. The enzyme activator is called enterokinase. This enzyme activator is converted in trypsinogen to trypsin. By the process is called autocatalytic mechanism process. That autocatalytic mechanism process will be completed after remaining enzymes will be self-activated. What is that? Chemotrypsinogen. It will be activated chemotrypsin and trypsin will be acting proteins. That proteins will be converted into tripeptides and dipeptides with the help of carboxypeptidase and aminopeptidase. Aminopeptidase means the proteins and peptones and proteases. Three things are to be converted into dipeptides and tripeptides with the help of carboxypeptidase and aminopeptidase. And conversion, trypsinogen and chymotrypsin will be converted into dipeptides and tripeptides. Okay, and one more thing is there. Next, tripeptidase. Tripeptides, not days. Very important. Tripeptides are peptidase. It's very important. The tripeptides is tripeptides. It is to be converted into dipeptides and amino acids. Dipeptides and amino acids. with the help of tripeptidase enzyme. Peptidase enzyme. Here tripeptides with the help of tripeptidase enzyme to converted dipeptides and amino acids. Dipeptides and amino acids. Next, one more thing is dipeptides. Dipeptides converted into with the help of dipeptidase enzyme. Dipeptidase enzyme converted into amino acids and amino acids and amino acids and amino acids. Okay, amino acids and amino acids. Okay, the protein digestion will be completed. Okay, tripeptides and dipeptides. Okay, alright. Okay, now we will start. So, carbohydrate digestion. Okay. Next, carbohydrate digestion. So already we will discuss where in our mouth starch or carbohydrates. Conversion of which enzyme? Amylase or ptyalin. The conversion of what is that? In the form of, already we will discuss, maltose, sucrose, lactose. So in the form of disaccharides. Disaccharides means it is a sucrose, maltose and lactose. Carbohydrates will be breakdown sucrose, maltose, lactose. We will see sucrose, maltose, lactose, how it will be digested. Okay, next, first thing is sucrose. Sucrose is converted into sucrase enzyme. Glucose and fructose. Okay, the sucrose will be breakdown. Who is conversion? Sucrase Sucrase enzyme means glucose plus fructose. Glucose plus fructose. Next, maltose. The maltose will be converted in maltase enzyme. Maltase enzyme. It is also to be breakdown into glucose and glucose. 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 Next, lactose. Next thing is called lactose. The lactose will be conversion is lactase enzyme. Lactase enzyme, and that is to be converted into glucose and galactose. Glucose and galactose. Glucose and galactose. Glucose and galactose. Okay, this is the story about disaccharides. This is the story about disaccharides. Okay, this is also, this enzyme also to be invertase. This enzyme also to be called as invertase enzyme. This is also called invertase enzyme. This all breakdown is directly going to villi. Directly going into villi. Okay, lactase enzyme is coming from villi. Okay, these are directly going into villi and directly to absorption. The carbohydrate digestion will be completed. Next, we will see now fat digestion. Fat digestion. This is called carbohydrate digestion. Okay, right. Fats or lipids. Fats are lipid digestion. Okay, we are discussing the fats. First will be where it will be breakdown means. Okay, the liver is secreting bile juice. That bile juice is not containing enzymes. It is also called a complex molecule. Okay, right. It is a complex molecules. Okay, bile juice not containing enzymes. It is bile juice. It is to be preparing for fat digestion. Okay, the bile is preparing for fat digestion. Preparing for fat digestion. That process is called emulsification. That process is called emulsification. Emulsification means okay, this emulsification means that fats means the bile going to work, or to coated fats, bile going to work are coated. Bile coated fats. Bile is coating for the fats. Bile is coating for the fats. After the lipase enzyme will be working. After lipase enzyme will be working. After lipase enzyme will work. Okay. The bile juice is not containing enzymes. The bile juice will be releases, to preparing for fat digestion. That process is called emulsification. Emulsification means that bile juice will be coated into the fats. So whenever fat coating will be completed, then lipase enzyme will be work. So where it will be work? Bile coated fats. After lipase enzyme will be work. Lipase enzyme will be work. That is called triglycerides. This triglycerides is converted into diglycerides. Help of lipase enzyme to convert it into triglyceride to diglycerides. These diglycerides converted into with the help of lipase enzyme monoglycerides. That monoglycerides are to be converted into two forms with the help of lipase enzyme. It is called fatty acids and glycerol. Fatty acids and glycerols. Okay. Fat or lipid digestion. The fat or lipid digestion, here bile juice. The bile juice is not containing any enzymes. The bile juice will be preparing for fat digestion. So that process is called emulsification. Emulsification means the bile is going to coated the fats. Then lipase enzyme going to work. Whenever the bile will be coated into the fats, for example, this is fat molecules, so it will be coated in bile juice. Whenever bile juice will be coated in the fats, then lipase enzyme will be work. It is in the form. This is emulsification. After enzyme, lipase enzyme will be working triglycerides. That triglycerides are to be converted into diglycerides in the help of lipase enzyme. That diglycerides are converted into monoglycerides with the help of lipase enzyme. Monoglycerides are to be converted into fatty acids and glycerols in the help of lipase enzyme. Finally, the fatty acids and glycerols will be digested. Absorption. It is called absorption form. Okay, absorption form. Right. Okay, this is fats and lipid digestion. Fats and lipid digestion. Okay, right. Next, coming to next one is nucleic acid digestion. Nucleic acids digestion. Nucleic acids digestion. Nucleic acids digestion. Nucleic acids digestion. The nucleic acids is having. The nucleic acids is having. So the first guanine 2, it is double bond. It is to thymine and guanine to thymine, and one more thing is, what is that? Sorry, first alanine to double bond and thymine. Next guanine to triple bond, it is called cystosine. Next, here it will be connected sugars, means ribose sugars and deoxyribose. Then finally it will be attached in phosphates. And here also there. What is this here? Sugars, and finally it will attach in phosphate. How it will be digested? First, it is a double helical. First, it will be break to the single helix. It will be divided into two helixes. So how it will be break the bonds? Now we will see that which enzyme is to be working to break the bonds. We will see now. Okay, the first breaking the bond, it is called nuclease enzyme. This bond will be break. The first breaking bond, it is double helix. It will be divided into single helixes. Which enzyme is to be breaking? Nuclease enzyme. It is to be first break the single helix. Two helixes are divided. Next, coming to the second. First, this is called breakdown nuclease enzyme. Nuclease enzyme. Okay, please observe. Okay, the bonds are to be breaking two things. Means double helix model. Okay, it will be breakdown into single helix. Next, second thing is it will be breakdown into phosphate and sugars. It will be breakdown. Here which enzyme is to be working? Nucleotidase. These three are called tidase. Means these all are called tidase. Okay, this nucleotidase enzyme means phosphate, sugars and molecules it is to be attached. It is called nucleotides. These nucleotides will be, first helix will be breakdown. Then after this phosphate, sugars and and alanine and guanine. It is called nucleotides, and three bonds will be first. This is to be second. It will be this is first breakdown. The helix will be breakdown into single. So next phosphate, sugars, and alanine and guanine. So here three bonds are there. This is called nucleotides. Means three bonds are to be combined. It is called tides. Here it is to be phosphate and sugar. In between the bond will be break. The enzyme is called nucleotidase enzyme. Nucleotidase enzyme. Next, sugar and molecules between sugars and molecules between this bond also to be break. This bond also to be break. Which enzyme is to be here working to break? Nucleosidase. Here only two bonds. Here only two bonds. Here it is to be working nucleosidase. Nucleosidase enzyme. Nucleosidase enzyme. Okay, this is to be breakdown in single bonds. Then after absorption will be started. In these two enzymes. These two enzymes are released in intestinal juice. Intestinal juice means succus enterica. Intestinal glands are to be releasing in intestinal juice. Intestinal juice is called succus enterica. Are you understand? Okay, the nucleic acids has to be formed and triple bonds. Triple bonds means three. One is phosphates, nitrogen fixation, and sugars. Okay, these three are combined. It is called nucleotidase. Nucleotide means three bonds are combined. It is called nucleotide. And sugars and nitrogen fixation will be combined. That is called nucleoside. Nucleosides means only two bonds are combined. Nucleoside. So here we will break that bonds. First helix, double helix. Every DNA will be formed or nucleic acids will be formed double helix. First we can divide the helixes. After we will break the bonds. First break the helix. After we will break the bonds. First phosphates and sugars in middle bond will be break. Sugars and molecules, the nucleosidase enzyme will be work to break that bonds. Okay, the nucleic acids digestion will be completed. Next, we will see villi. Next we will see the villi. Next we will see the villi in absorption of food. Absorption of food in villi. Absorption of food. Right. Okay, we will draw the diagram. Okay, are you observing this diagram? This is called villi. This is called villi. So here it is to be having epithelium. Here it is to be having epithelium. It is called breast border epithelium. Okay, this border is called epithelium. Are you? Epithelium. Okay, everyone should draw the diagram and write one by one to understand clearly and easy. Clearly and easy. Easy method. So in middle, this is called lactile. This is called lactile. This is called lactile. This is called artery. This is called artery. And this is called vein. This is called vein. This is lacteal. This is lacteal. And this is villi. This is villi. This is called epithelium. This is called epithelium. Artery, vein, and lacteal. Lacteal nothing but lymph node. It is nothing but lymph node. Lymph node. It's nothing but lymph node. Okay? Are you understand the diagram? Are you understand the diagram? So means in intestinal walls. In intestinal walls you have to see, this is the intestinal walls. Here brush border like. Okay, brush border is there. This is called microvilli. This is called villi. This is called villi. We will see big diagram. We will see the big diagram in how to diffuse that food particle to inside. Food particles to inside. Clearly to understand. Everyone, you can draw the diagram and one by one you can see and easily to understand. Easily to understand. Okay. The first thing is called glucose, galactose, and amino acids. Glucose, galactose, amino acids. Amino acids. These three things with the help of sodium ions, that is called a carrier ions. That is called carrier ions. Okay, glucose, galactose, and amino acid with the help of carrier ions. Okay, that is called sodium ions. To be secondary, what is that? Secondary transport diffusion. It is called secondary active transport. The help of secondary active transport. What is that? Glucose, galactose, amino acids with the help of carrier ions. help of carrier ions in secondary active transport. Secondary active transport. Here will be after this membrane will be. So enter will be inside. Entered will be inside. Enter will be inside. Means this is the wall. This is the epithelium. So help of secondary active transport, it will be diffused into inside. Here it is having. Here it is having to insert into the blood vessels or blood. Okay, they are going to diffuse blood. So which transport here? It is called, which stands for facilitated transport. Facilitated diffusion means facilitated diffusion. Facilitated diffusion. Are you understand? Glucose, galactose, amino acid. Glucose, galactose, amino acids. Glucose, galactose, amino acids with the help of carrier ions. That carrier ions is called sodium ions. Sodium ions. It is to be that is moving for secondary active transport to inside. So inside to going to blood. Inside to going to entering into the blood. It is called facilitated diffusion. Facilitated diffusion. Facilitated diffusion. Are you understand? So in case any doubt, okay, please observing and repeat again, you will see. Repeat again, you will see. And the second one is called fructose. And the second thing is called fructose. The fructose will be diffused inside. It is called facilitated diffusion. So inside to blood, it is also having facilitated diffusion. Facilitated diffusion. Facilitated diffusion. Next, water. Next, water. Water is going to diffuse inside osmotic gradient. Osmotic gradient. Osmotic gradient. Osmotic gradient. Water will be diffused to inside to blood. It is also having osmotic gradient. Osmotic gradient. Okay, inside also to diffuse osmotic gradient. Osmotic gradients. Next, fourth one. Okay, fourth one will be diffuse small fats. Small fats. Small fats. Small fats will be diffuse. Small fats will be diffuse. Means by using diffusion process. Small fats are using to entering for the blood. Entering for the blood. It is also called diffusion process. It is also called diffusion process. Diffusion process. Diffusion process. Next thing is called large fats. Next thing is called large fats. Entering into the large fats. Okay. Large fats. Large fats are not directly to enter. Not directly to enter. Not directly to enter. With the help of bile juice. With the help of bile juice means the large fat is converted in. With the help of bile juice is called micelles. Micelles. Okay, micelles. This micelles going to enter the epithelium. This micelles going to enter the epithelium. going to enter the epithelium. This epithelium was coated. This epithelium was coated. This epithelium was coated with proteins. This epithelium was coated with proteins. Coated with proteins. That proteins. Okay, that proteins is called chylomicrons. That proteins is called chylomicrons. These chylomicrons will be entering into the Okay, here the epithelium will be coated. Here it is to be coated. And here it is to be coated proteins. Coated with proteins. That is called chylomicrons. Chylomicrons is also called exocytosis. It is also called, it is called exocytosis. Exocytosis. The microns will be entering into the lactee. What is called? Chylomicrons. Okay, the large fats are not directly to enter with the help of bile to convert into micelles. This micelles going to bind with epithelium. That epithelium will be coated with proteins. That is called chylomicrons. That chylomicrons will be entered into lactee. In this lactee, the chylomicrons will be entered into subclavian vein. Next, subclavian vein to thoracic duct. Next, thoracic duct to blood circulation. After entering into blood circulation. After blood circulation, the wall of blood vessels. Is having endothelium. Okay, endothelium. Okay, this endothelium will be releasing lipoprotein lipase enzyme. Lipoprotein lipase. This lipoprotein lipase, it will be working on chylomicron. These chylomicrons will be converted into two forms. That is called fatty acids and glycerol. Fatty acids and glycerols. Okay, this is the story about absorption of food in villi. Food in villi. Food in villi. Okay, the large fats are not directly to enter into the blood circulation. Remaining all things are to be entering into the unfacilitated diffusion and secondary active transport and diffusion process and osmotic gradient process. They are directly through enter into the blood circulation. But large fats are not directly entering into the blood circulation. Okay, because they are not directly to absorption form, so they will entering into the lacteal. They will entering into the lacteal. That is called chylomicrons. That is called chylomicrons. Okay, large fats with the help of bile, to convert it into the micelles. Converted into the micelles. That micelles going to bind with the epithelium. That epithelium will be coated with protein. That is called exocytosis. That coated with proteins is called entering into the lacteal. That is called chylomicrons. Okay, here also coated protein with, it is converted into chylomicron. This process is called exocytosis. The chylomicrons will be entering into the lacteal. This is also called chylomicrons. Here, once coated with proteins, they will be converted into chylomicrons. The process is called exocytosis. They will be entering into the lacteal. Here lacteal is having, here chylomicrons will be entering into the first subclavian vein to thoracic duct to entering into the blood circulation. After blood circulation, the wall of blood vessels is having endothelium is there. That endothelium will be releasing lipoprotein lipase enzyme. That lipoprotein lipase enzyme going to work chylomicrons. That chylomicron will be converted into fatty acids and glycerols. And also fatty acids and glycerols to be stored on adipose tissue or liver in the form of triglycerides. Okay, these fatty acids and glycerols will be stored in adipose tissue or liver in the form of triglycerides. Okay, this is the absorption of food in microvilli. Okay, are you understand? Right. Okay, now we are moving into the role of hormones. Once you will see and check everything. Okay, 2 minutes or 1 minute. Observe please in this diagram. It is very important. This is finally the breakdown food particles will be diffused into the blood circulation. So then directly going to reach the cells. Okay, you have to see here. This is called artery. This is called vein. In middle it is having lacteal. Okay, so micelles will going to bind with the which epithelium? That epithelium will be coated with protein, that is called chylomicrons. This process is called exocytosis. The chylomicrons will be entered into lacteal. Okay, lacteals will be entered into subclavian to thoracic region to after entering into the blood circulation. So the wall of blood vessels is having endothelium. Endothelium is releasing a lipoprotein lipase enzyme. That enzyme is going to work chylomicrons. That chylomicrons will be converted into fatty acids and glycerols. These fatty acids and glycerols will be stored in adipose tissue or liver in the form of triglycerides. Next thing is called role of hormones. You can observe, very important. The first one is called, please observe the role of hormones. The first hormone is called gastrin. Next, second one is, what is that second hormone? Enterogastrin. Next, third. What is the third? Next one is called secretin. It is very important. It is very important, secretin. Next, fourth one is called cholecystokinin. It is also called pancreozyme. Next, fifth one is called enterocrinin. Next, sixth one is called villicrinin. Next, where it is to be releasing and what is the function? So gastrin is releasing. Where the gastrin is releasing? Stomach. Where? Pyloric part. What is the action? Stimulates secretion of all gastric enzymes. Next, what is the action? Stimulate to secretion of all gastric enzymes. Next, enterogastrin coming from duodenum. What is the action? Inhibit the gastrin, inhibit the emptying of stomach. Inhibit the gastrin and inhibit emptying stomach. Next, secreting. It is very important. Secretin works on pancreas. It is also to be releasing in duodenum part. To works as a pancreas. To works as pancreas. To secretin is very important in our intestine. Okay, works on pancreas to release water and bicarbonate ions. net ions. Okay. Why? To works as pancreas. Okay. To release water and bicarbonate ions. Why it is releasing means the stomach is having highly acidic medium food, that is called partially digested food, that is chyme. That is chyme. So that chyme will be neutralized. That chyme will be neutralized to convert alkaline. Means not converted, it is neutralized to alkaline medium. Means acidic medium to alkaline medium. Alkaline medium. The secretin, it is very, very important. Secretin is very, very important. Very, very important. Okay, to work as pancreas, to release water and bicarbonate ions. Bicarbonate ions. That two things have to be neutralize the chyme. Neutralize the chyme to alkaline food. Chyme to alkaline food. Chyme food to alkaline food. Next cholecystokinin. Next cholecystokinin are pancreozyme. This is also to be released duodenum part. Next works as a next means works on pancreas. 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 Next, pancreas and to releasing enzymes. To releasing enzymes. To releasing enzymes. To releasing enzymes. Next, contraction of gallbladder and relaxation of OD sphincter. Release enzymes. Release enzymes and contraction of gallbladder. Next, contraction of gall bladder and relaxation of OD sphincter. Relaxation of OD sphincter. OD sphincter. So in these two it is to be releasing bile juice. Releasing bile juice. Releasing bile juice. Bile juice releasing. The contraction of gallbladder means it is to be contraction. Means the bile will be comes out. Next, relaxation of sphincter. Mean it freely will open. Though bile juice will be releasing into the duodenum. Bile juice will be releasing into the duodenum part. Duodenum part. Okay. Next, enterocrine. Enterocrine. What is the role of enterocrine? Means stimulate secretion of succus enterica. Stimulate what is that? Stimulate secretion of succus enterica. Secretion of succus enterica. Next, villi crinin. Stimulate the absorption of villi to improve absorption. Stimulate villi to improve absorption. Okay, this is the role of hormones. Okay, to observe all things. the digestion and absorption topic will be completed. Digestion and absorption topic will be completed, and tomorrow we will discuss a new topic. Okay, you can observe all the things. The role of hormones means gastrin, enterogastrin, secretin, cholecystokinin, enterokinin, villikinin. Here very important is called secretin. Secretin works as a pancreas. Secretin works as a pancreas to release water and bicarbonate ions to neutralize the acidic medium food to alkaline food. It is very important. Okay. Next, cholecystokinin also to be works as a pancreas, releasing enzymes, and it is a contraction of gallbladder and relaxation of OD sphincter. That's time the bile juice will be releasing. Next, enterokinin and villikinin is there. Okay, right. Okay, thank you. Tomorrow we will discuss new topic.